Okay, good. Fine. So, after that, shall we play a game? It's about, I think your presentations start in a month from now, April, that week of April 15th, whatever that Monday is, right. So, we will have to choose the order in which you present, because it is spread over 2 weeks, and people will complain it is unfair that you go first and you go first and so on. So, we will leave it to your luck, right. So, this is what I typically use, it works fine. Earlier I used to pick chits and so on and so forth, tear pieces of paper, write numbers and so ask somebody to draw lots and then choose that. And then somebody about 5 years ago suggested that I use an online tool, which made sense. So, from that time onwards I have been using this tool, which works well. So, let me, there are 11 in class, 11 according to serial number. Okay, serial number goes as, for purposes of full disclosure, serial number 1 is Sujitra, it goes as alphabetical order of roll numbers. So, BT 23, B029 comes first, and then D comes, and then M comes, okay. And then B's come, and then ME comes. So, ME is the last, that is the way it goes. So, whichever number comes, I will start entering, and that will be the order in which you will present. Okay. We will have 2 per day. So, let us see who is the lucky person. Smile here. Number 1, all right, Sujitra presents first, great. So, 1, okay. 1 would, okay, how should I do this? When are the dates? Okay, I will write down the numbers, and then, no, that won't work. So, I need the dates. So, it starts 15th April. So, this one is 15th April. So, the next one is also 15th April, right. So, who is the next lucky one? Number 6. Number 6 happens to be 7 here, Manisha. All right. Good one. Okay, just a second. So, Manisha goes on the first day. April. Okay. The next date coming up is, next spins, couple of spins are for 18th April. Okay. So, 18th April, who is the first one to go? So, this 1, 2 is also important. No, well, we can change, but 15th April A, 15th April B. Okay. April B. Okay. So, that way I have complete information. Number 10. Number 10 is Vikash. All right. Vikash is happy. So, at least he is not the first. So, 18th is it? 18th April A. Okay. So, this will also be 18th April A. No? Okay, I will take care of that later. So, now remove choice and we spin. Manisha is all smiles, she is done. Good. 3. It was deciding between 3 and 4, 3. 3 is Jayashree. 3 is Jayashree. So 18th April B. Okay. So now 2 for 19th April. All right, 11. Dr. Lakshmanan. Okay. 19th April A. Okay. Then spin. 9. 9 would be Swati. All right. That's a nice date, somewhere in the middle. So 19th April B. Okay, so 5 more remaining. I didn't tell you the date. This is Monday 22nd. Number 5. 1, 2, 3, 4, 5. Jashwan. All right. Jashwan is 22nd. 22nd April A. All right. Then next is 22nd April B. Seven. 1, 2, 3, 4, 5, 6. Praveen. So this is 22nd April B, is it? Okay. Okay. Then only 3 more remaining. Not bad. One more choice. Who are the 3? 1, 2 and okay. So 3, the next date is 25th April. So yeah, only this thing is a holiday. Yeah, 25th March is a holiday. Good. 25th April is Thursday. First person. Eight. 8 is Raghavi. Okay. So 25th April, is it? Okay. And last spin. 25th April B. To Ankita. Ankita, 25th April. April B, is it? Okay, sorry. And so the Abbas gets special treatment on 26th April. Lone person presenting. Yeah, Friday. No, 26 is Friday. Friday. Monday I have as a buffer, just in case some holiday gets declared, then we push the whole thing by a day. That's the way it works. So, the thing is, you should all ask questions. You'll be graded for asking questions. Okay, none of you had been in such a class earlier. Okay, so there'll be enough incentive. The way it will work is, the person presents, comes here and presents, gets the stage, and then all of you ask questions. I will keep marking who is asking questions, and the questions I put them into 3 boxes: routine, medium level, and in-depth questions. Okay, so a judicious mix of all 3 would be good over the entire period. So I look I'd take all the 11, 10 presentations into account for each person, because you're on presentation, you won't ask yourself questions, and then I will average them out. As long as you ask a minimum, you'll get those 5 marks, the 5 marks set aside for that, right, 10%. So that's participation, whatever the marks are, I don't remember exactly, it is, but it's a reasonably good number to give you incentive to ask questions. So please ask questions, feel free, that way we learn. By the way, the person presenting is not an expert, therefore the person may not know all the answers, that is perfectly fine. The best strategy is make sure that you know enough so that you do not embarrass yourself. At the same time, there are things that you do not know, right. Even with 30 years of experience, you may not know certain things. So then it's good to accept and say, I'll get back to you, or you can discuss later, and so on. So the questions are asked in the spirit of knowing and so on and so forth, not to put anybody down. Okay. So that is always better. And yeah, I think this is a reasonably well knit class. I didn't see any major tussle, or at least I didn't notice any major tussle. So I think this is a reasonable, I don't expect major problems in this class. And I can sense problems from a distance. When I sense, I will get in and set it right. Don't worry about it. So even if you have somebody who is politically against you, I'll take care of. Politics, unfortunately, I cannot get rid of. So I hate politics. I hate politics and everything. I think that spoils the whole thing. So, but I've been in administration, by the way. I've set up 2 departments and all that. I know. I've been in the thick of things. Okay, so that's good. We had fun. 26th April, lone person. Okay. So I've noted down things. So this is done, and I've opened up the next set, which is reactor species and other stresses. Right. There are 4 papers. So let us go through. Tomorrow is off, I mean, for a reading. If you want Monday also off, we'll think about it at the end of this class and then see, depending on how this class goes and so on.
So we don't have class tomorrow.
Tomorrow there is no class. It's no class in the sense it's for reading.
Oh.
So some time I need to give you for reading, and that too a larger number. I typically start out with 1, but this time I decided to do that directly rather than a reading exercise. And then it went to 2. You started with 2 papers, and next time it became 4 papers, and this time also it's 4 papers. So you need some time to internalize that and so on and so forth. So at least I am giving you time. Make make judicious use of that. Okay. So coming to this, before we start, any of your comments? Why don't we start with your comments? That might be better. I have something to, essentially the same thing to say. Some stories and so on and so forth, or probably some stories may come. But instead, if you have questions based on the papers, otherwise I'll prime you. So these set of papers were on using reactive species induction to improve bioreactor productivities. We have done work on quite a few systems, but we have published on only a few systems. So I've given you the papers that we published, and some were optional anyway. Right. So any general comments, questions, or specific also?
Is there any reason why there was no change in morphology of A Niger when you treat it with HOCl H2O2 and you leave it untreated?
For the morphological changes, it has to there has to be enough differences in the things that constitute its cell wall, cell membrane, and so on and so forth. So that is my guess. They are not majorly affected, and not just that, morphology is quite a complex thing. You may have read in one of those papers, no, not coming up, look at Susmita's paper. This was bacillus. Bacillus is rod shaped, right. So we looked at bacillus that were grown in under standard conditions, shake flask conditions, and with shear of 1482 second inverse. shear rate of 1482 second inverse, 1500 rpm, the maximum rotation speed of that particular device, a shear co-wet flow device. Okay. We found when you shear the cells, you expect them to elongate, right? That is our intuitive expectation. What we found was that actually they contracted. Okay. Then we reasoned it out as probably it was trying to overcome the stress effects, and therefore, the opposite processes were set in place and it kind of contracted and so on and so forth. So, that is a rather complex kind of a phenotype to comment on easily, the cell shape and cell morphology. And with Xanthomonas, it sometimes it produces gum and sometimes it does not produce gum. So, that is called mucoidy, and that also is quite a complex thing, because it uses antioxidant for many different aspects, including fighting against reactive species, against a plant pathogen and so on. So, it is a plant pathogen. A reactive species of the plant, when it is trying to destroy it, when it is attacking the plants, then it uses that. So, very interesting things came about, highly reproducible. So, that is the best that I can tell you in terms of those. We do not understand it, I mean we did not even look at it. So, I should not even claim to do that. I have not seen any studies, but my contact with or active contact with that literature probably moved away from it about 20 plus years ago. So, if somebody had looked at it and figured out reasons, if we come across that, please share that with us. That will be good. Any other comments, questions? Okay, yeah. Ask later, okay. Fine, I'll prime you up. So, this is where our story ended last time. With LPOS, the xanthan gum productivity was higher, but the maximum cell concentration, even though there is not much difference in the growth rates, the maximum cell concentration with LPOS was about half of that we got through the normal cultivation or the air cultivation with air and so on and so forth, the control cultivation. Okay. So, our thinking was if we can get this up to here by some means, then we have much better productivity, each cell producing more and more number of cells, and therefore, overall better productivity. That was the motivation. And we kind of knew that the reactive oxygen species are causing this when you add hydrogen peroxide. The reaction itself is quite green. It produces the catalasic decomposition of hydrogen peroxide produces only water and oxygen, right. Whereas iron and other metals are present throughout. We use tap water because it needs iron and so on and so forth. So, iron is present, definitely iron is present, maybe other metals also. When iron is present, Fenton reaction will definitely take place, and that can produce hydroxyl radicals, and that can destroy cells. Right. So, we suspected that to be causing this difficulty. So, this was the scene when Manjula entered, and she asked the question, can I vaccinate? It was her idea completely, and this was her idea in the first 15 days of her being in the program. Okay. Jan 1996 she started. One of the first conversations was that can we do this? She was an amazing person, amazing in terms of work, but I would never recommend her work culture to anybody. We immensely benefited from that. She is one of the first students. Many of us in the lab were young. Venkatesh was young. Venkatesh joined 3 months after me, and 2 of us were there till the 3rd person joined much later, and so till Pramod joined much later. Yeah, Pramod joined many years later, 3-4 years later. So, both our students were working. This one would work through the night, bring in sleeping bags to sleep in the lab without me asking for it at all. And that was a very infective, what shall I say, infective culture. So, everybody started doing that. The productivity of the lab went up and so on and so forth. And I had to kind of calm her down. She was a married person. Okay. She kept her husband somewhere else, and well into this. He would come and visit, and I had to force her to take a break, go spend time with your husband, and then come back. That kind of a thing. It was funny. At one point in time, I didn't know who was the guide and who was the student. So during the entire, she submitted a thesis in 2 years, 9 months with 5 papers. So amazing, really amazing. But I wouldn't recommend that work style to anybody. She was a mad person really to work that way. And she read a lot, huge amount of reading. Okay. So she asked, can we vaccinate? I said, yeah, of course. Why don't we try this? I didn't have any ideas of my own at that time. We knew that reactive oxygen species, and we had to look at it, investigate, try it out. So that's how we entered this field of reactive species in 1996, or before that, by recognizing that we need to address it. Before you actually do this in a vaccination sense, you sensitize the cells, and then you grow them in LPOs. So the cells are supposed to take it better. That was expectation. However, this is a scientific study. So what does vaccination do to the cells? What does the exposure to a reactive species generating agent do to the cells when it is grown under normal conditions? We needed to check. So essentially characterize the vaccination effects. So when we did that, we were in for a surprise. Okay. This is the growth curve, cell concentration versus time. Both were grown in aeration, one without treatment and the other one with treatment. Okay. The wild type is a misnomer. If you say wild type, biologists will jump at you. So please call this untreated. Okay. Untreated, and engineers are fine with this, but untreated and HOCl treated. Okay. So not much of a difference in growth rates, not much of a difference in the maximum cell concentration. So the treatment was fine. She had used a 3-stage treatment, which I think she adapted from Tuati and Dukan and Tuati paper, which looked at the effect of HOCl on water cells involved in water treatment. Right? And I think they used a 3-stage. She adapted it to this, and then she used it. And the major surprise came with xanthan gum. The 50R xanthan gum was up more than 2-fold, right? Just treatment of the inoculum with HOCl to induce hydroxyl radicals increased the volumetric xanthan gum level more than 2-fold. Okay, this is what matters. This is what makes you money, makes an industry money. Not just that, it was of higher quality. This is a significant improvement in quality of the viscosity. Standard conditions, the standard conditions are set. What shall I say? She isolated the gum and then dissolved it in appropriate water, to get appropriate concentrations, and did this test. And then it is known that if you have more pyruvate, the quality of the gum will be less. If you have more acetate, the quality of the gum, if you have more pyruvate, the quality will be more. If you have more acetate, the quality will be less. Quality in the sense the viscosity that the gum can create, and that was shown because xanthan gum is glucose, glucose, glucose with a lot of side chains and so on and so forth. Some of these side chains are pyruvylated, some of these side chains are acetylated. So more the pyruvate, better the quality. That was known at that time. So she did analysis of the gum using one of these high flown techniques and mass spectrometry, so HPLC or something like that. And then she came up with, we found that its pyruvylation was higher, acetylation was lower, and so that ties in with this. So you get a better quality gum and much higher quantity gum just by treating the inoculum with RS generating agent, HOCl in this case, a reactive species generating agent, a hydroxyl radical generating agent. Okay. So as soon as I saw the result, I said, if my aim is to improve productivity, it's improving quality as well as volume here. So why not use this as a strategy? So we started trying, and at that point in time, the 15 systems that we tried, it worked. Okay. So we tried it on some of which we published, right? And these are all the published things with different systems giving higher productivity just by treating it with the inoculum. We understand it to a certain extent as to why this happens in the case of the xanthomonas system. The other systems we didn't study, xanthomonas system. So that is we have already seen some details. The SOX, the reactive oxygen species activates the SOX RS regulon, which consists of the SOX S, SOX R and the SOX S genes, proteins rather, SOX R and the SOX S proteins, whose levels remain high in subsequent generations as an adaptive response. Not if you treat it with those doses of HOCl, the levels of SOX R, SOX S go high, and they somehow remain high without any change to the DNA. There is no mutation, okay, because the levels remain high. The levels remain high for about 14-15 generations, and then they come down, okay, come down to reasonably low levels. So it is only for that. So which means that the DNA is not getting affected. It's a known case of adaptive response. It's called adaptive response in the literature. The exact reason is not known. SOX S binds to the strong promoter of the GUM-B gene, and SOX S levels are high, therefore you get more transcript levels. Till here we had proved, right, that its binding we had proved, its high levels we had proved, and so on and so forth, but not the translation part. So we expect the translation part to have been higher also to produce higher GUM. We see higher GUM definitely. So that part we didn't prove, but this leads to higher mRNA production, which could further lead to, which leads to further GUM production, most likely through better translation. Okay. So that is the mechanism. Also, if you plot the specific xanthan gum level, that is the amount of xanthan gum each cell produces versus the amount of reactive species that each cell experiences, there is actually an increase in this region. In this region of reactive species, we called it free radicals at that time. Okay, this was more than 25 years ago. So there is a region in which it increases and then it decreases. The decrease part was well known, right? It's the negative effects of the reactive species, whereas actually it going up was not known. This is something that we found, and the optimum in this case happens to be 0.33 millimole per gram cell. Then with the help of K Suresh, my colleague there, we built a model from first principles. This is the reason for presenting the second chapter as initiation, termination, propagation and termination. Okay, it helps. So we looked at the initiation reactions, termination reactions, and then invoked the pseudo steady state approximation to equate the initiation rate with the termination rate, and that simplified our analysis. And also these relationships, we came up with generic relationships, found these constants. We used some data to find these constants and then use the other data to verify whether the model works or not. So these things were actually one set, I think was, hold on, I think this and this were used for coming up with the constants, this data, and then we used those constants and we saw very close results to experimentally found results for growth rates. I think no, this is Ross level, sorry. This is the reactive oxygen species levels when no treatment was done, when only H2O2 was used. H2O2 was used with vitamin C, H2O2 was used with vitamin E, and both the antioxidants were used with H2O2. Okay. So the model seems to work well. All this was published, and that is all I thought I will present for you to initiate any discussion.
So this term quality, here we understand because of the gum we recognize the viscosity. In general terms, when you talk about the quality of it is improving the quality of the bioproduct. What does it mean usually? So how is the quality improved? Are we talking about the structure being better?
It could mean different things in different contexts. What is quality of a person? What is quality of work? Okay. What is quality of an art piece? Right. There is a certain level of, I mean in these cases there is not much qualitative aspects to the quality part of it as an art, but it still is. People who look for very high quality things are typically alone because the population will not understand it. That is a typical thing. So, in the case of gum, higher viscosity was looked for at lower levels, lower concentrations, that is quality. In the case of a drug, probably a lower dose that is producing the same effect without much side effects is a higher quality thing. Okay, I would include the side effects also, definitely, after all the experiences that I have gone through. So, it is, what is quality of life? Right. What is quality of life? Just being alive? I do not think so. Our doctor definitely will not agree. So, it is being able to do normal life activities even though you are hampered, rather you could be hampered. Right. How well are you able to get back to normalcy after a heart procedure maybe? Yeah, they talk about quality and morbidity in some sense. So, it is different in different contexts, it is a higher level of operation as far as I see it. I look for quality, by the way. I have been badly affected by lack of quality, but I cannot talk about it in a population.
What is the use of main use of xanthan gum?
Xanthan gum is used in many different products, foods. It's a viscosity modifier. It's a basic viscosity modifier, heavily used viscosity modifier. So, you will find them in gums, you will find them in squashes, you will find them in so many different things. It is used heavily. There were a lot of enquiries when somebody knew that we were working with xanthan, and that period when we were working with xanthan gum. Even after I came to IIT Madras, somebody called me to talk about xanthan gum. So, I guess they were looking for some ready-made process that I could give them and so on and so forth. No, we had looked at reactive species. So that's more of a philosophical question. If you don't, suppose you are looking at some constructions, something that looks elegant and lasts a long time is of high quality, in my opinion. Okay. Many people would not want that even. I mean, contractors would just bulldoze, get away with the minimum possible things. So, they are not looking for quality. Okay. We built a house, went through hell rebuilding the house, and tussle with the contractor, because the contractor wants to get things done the minimum possible thing. So, he said, I asked him for Legrand switches because I had heard that they are good and all that. So, he agreed, and then he called me for the shop and badgered me saying that Litaski is good, so you put that and all that. Okay. I said, if it does not work, you will have to change the whole thing. Okay. I mean, I tried telling him, no, it is not good. The reviews are not good and things like that, but he insisted because that would reduce his cost by 1/3 or something like that, or at least half. So, on that condition I took. Before we occupied the house, right, 2 fan regulators went within a span of about a month and a half of not much use. They would have probably tested it and so on. So, switches where it will go like this inside, okay, no clean thing. So, then I caught him and said, you better change it, otherwise he had to change at least in one of our units. And so, that is, so it is context based at best.
Also, xanthomonas campestris is what is published in majority of the papers, right?
Xanthomonas campus?
Majority of the papers are with this system.
For the LPOS, yes.
Yeah, for the LPO.
For the LPOS, yes, we have worked with many different systems. See, my initial thing was I expected this to be expensive, although it later turned out to be not in an effective sense. So I wanted some justification. So I said, at least if I choose a viscous fermentation, which is difficult to, you need much higher powers to provide the same KLa and so on and so forth, and which has industrial significance, it's a product. So when I look for both, this seemed appropriate. So that's the reason why we bought that strain and started the work. We started the work after we, yeah, that was when we got into LPOS itself. Right in the beginning we got this.
But the productivity increase was not what you expected in the first, right? In case that didn't happen, how would the rest of the
It would have taken a totally different turn. I probably wouldn't have gotten into reactive species at all. Rather, I wouldn't have gotten into reactive species as a lifetime occupation, research lifetime occupation. Probably would have looked at the LPO's relevance alone and moved on to something else. So that's why I say I'm lucky that I found my niche within 2 years of getting in. Many people, most people don't. Most people move around and then, so then I knew this is what I need to do. Although I had to make a sacrifice. It was not a popular area. I saw the richness in it, but very few people did. Right? They're not willing to work. Work on reactive species, research on reactive species has been on for at least 70, 80 years. But it is not a popular area. So if I get into that, all recognitions will not happen, funding difficult, student interest difficult unless I do something about it, which I did. I always showed the application of that to get the basic work done. So all that, I mean, it was all open, but this is what was given in the project titles to attract people. So all that had to be done and no regrets.
it just seems like a chance work in a sense.
Pardon?
You wouldn't have get the correct model system in the first go itself. In many cases, that's how it happens. We work with multiple systems and then fix on it and work. So here in Xanthomonas seems like.
No, I would say it works a little differently. I mean, maybe, I don't know. I would say being open to possibilities. For example, I could have been blinkered and said you treat it with with this thing and then we use or even when Manjula came in, when she asked me about vaccination, I said what are you talking about vaccination here, and gone on a different route and did some studies and so on and so forth. But to be open essentially and to recognize that ideas can come from anywhere as long as they are worth. And when ideas come, you better give them credit, right? That comes much later, by the way. And being open, having that spirit of adventure, having that spirit of exploration, that I think is more important. Okay, I was lucky to have that given my orientation. I could have done standard things and made sure my awards list was fine, my climbing list was fine, and so on. I had to wait 3.5 years before I could see a possibility of a paper. I had to wait 2 years plus to see the possibility of a funded project, whereas my colleagues were able to do that much earlier. Those things are part and parcel. The initial periods were tough, but yeah, but if you believe in, if you are able to believe in what you do, then it's okay. But the last 5 years I cannot do that. My students have to finish, so I changed. That I can do. I mean, and it kind of came to some sort of the adventurous part came to some sort of a natural conclusion with the reactive species module and the book. I wanted to write the book maybe starting 2028 or something like that, but it happened 10 years before that. I guess all for good. So now in a different area. Any other questions? Yeah. Last question, 10:50. Yeah.
So in this paper about your aspergillus cultivation,
Mhm.
large logically what should happen is when you induce it with HOCl and then use the LPOS strategy, your cell concentration should obviously
Go up.
be highest among the other treatments. But here, as we see, the group where the cells which were treated with HOCl, but they were aerated, they have like a very significant
Higher concentration compared to.
when you compare it with HOCl treated cells and cultivated using LPOS strategy.
Okay, I'll believe you. I haven't really looked at that data in this light, but good critical reading of the data there. If it is indeed true, the reason is it is not optimized. It was again done in the addition of hydrogen peroxide was done in a non-optimal fashion. We all the optimization was done for xanthomonas, and we got to a stage where there was 50% higher productivity and comparable growth and so on and so forth, Chaitali's papers. But these were experiments done in the lab. You needed a controller and things like that to take care of. We didn't have money for that at that time. So graduate students sitting in the lab and adding was how these experiments were done. So they were not optimal. If you optimize, I am sure the cell concentration would go up by minimizing the negative effects of reactive species on the growth part of it. But good reading. I mean, let me check the data. I haven't seen this data in this slide. You have cross checked. I mean, I am not surprised. That could very well be the case. But good reading, good critical reading. By the way, this is what we would like to see in a critique of papers. Okay, I haven't seen this. I haven't seen this. I am not surprised, but I haven't seen this at all. So that's very nice. Good. Okay. We'll have to stop here. What do you want to do on Monday? You want off or do you want off? Yeah, off is fine. No, I was prepared for off because I needed to give you time. I need to be fair. These things take time, right? You are reading it as a fresh person. You need time to internalize this. This is a part of the course. Fine. So we'll take off on tomorrow. There's no. It's a reading class. Monday is also a reading class. There are 4 papers that you need to 4 or 5 papers that you need to read. I don't exactly know. 4 is it? 4 papers that you need to read. So please read them well. This time is given to you for reading, and then learn, and then incentive. We'll have an incentive next Thursday. Fine.